Alright, in this video I'm going to go over the Hardy-Weinberg principles and also an example. Hopefully after we go through this graph here will make a little bit more sense. So starting with evolution and populations. Evolution is a change in allele frequencies, but hypothetically, what if conditions would cause allele frequencies not to change? So in a non-evolving population, there's 5 key factors that need to be considered. One, it has to be a very large population size with no genetic drift. There needs to be no migration in or out of the population, eliminating gene flow, and no mutations, so there's no change in the genetics. Random mating, so there's no selection of any individual or phenotype, and no natural selection that's occurring, meaning everyone is equally fit for the environment. There's no advantage to be one or the other. The Hardy-Weinberg equation is true only if these 5 things are met. So as I said here, terms and conditions do apply. Large population, random mating, no mutations, no migrations, and no natural selection. Rarely does this exist in nature, but this does allow this equation to hold true. So hypothetically, a non-evolving population preserves the allele frequencies. It serves as a model, which in this case we're going to call the null hypothesis. What a null hypothesis is this is referring to an area where if we have a hypothesis, we have a question, calculated mean for the sample. If there's a small difference, we fail to reject the hypothesis. That would be the null hypothesis there. If we do have enough data to reject the hypothesis, meaning it was wrong or we're proving it to not be accurate, in the case this would be a large difference, this results in the rejection of that hypothesis. So values that support the null hypothesis occur in the green region, and values that do not support the values supporting the alternate hypothesis occur in the red. Those are the extremes there. So natural populations rarely are in Hardy-Weinberg equilibrium. However, this is a useful model to measure the forces acting on a population. The null hypothesis means insignificant or no relationship between 2 variables. No difference is one way to consider it here. So the Hardy-Weinberg equilibrium, made by a mathematician with the last name Hardy and a physician with the last name Weinberg, came together, and they were concerned with questions like, what happens to frequencies of alleles in a population over time, and would you expect to see alleles disappear or become more frequent over time? And they're working with diploid Mendelian population here. So this population genetics is the study of properties of genes in a population. Genetic variation in a population puzzled scientists, and this was one way that they seek to try to explain it. So point out in large populations with random mating, allele frequencies remain constant. Dominant alleles do not in fact replace recessive ones as was once thought. So through the process of random mating, there's no advantage to any phenotype or genotype here. Here we have assortative mating, which is showing that there are, in this case, these bugs tend to mate with ones of the same size. This non-random mating here would be the same color would be mating together. We're talking about random mating, where this color bug will mate with any other color bugs with an equal chance. So here's getting to a little bit of an example here. We have a population of 100 tigers, to make it easy, and it's 84 orange and 16 white. So 84 out of 100 is really easy, it's 84%, and 16 out of 100 is 16%. However, because we're going to work with these in a math equation, we need to convert those to decimals, so we simply divide by 100. 84% is 0.84, 16% is 0.16. Remember, a population that is in Hardy-Weinberg equilibrium is not evolving. So the frequency. If there's not 100, you take the number of individuals and divide it by the total number of individuals being considered. In this case, it being 100 makes it very easy, but if this number was to differ, you take your individuals, divide it by the total. So let's get to an example. Assuming 2 alleles, big B and little b. The dominant allele is going to be, of course, big B. Recessive allele is going to be little b. However, in the Hardy-Weinberg equation, the dominant is going to be represented by this letter P, and the recessive by Q. So P plus Q, if we only have A and B, must equal 1 or 100%. And I have the examples here of the tigers that we're going to work with. I also have a couple links to some information about white tigers in another PowerPoint if you're interested in looking. So this is the Hardy-Weinberg equation right here. P squared plus 2PQ plus Q squared equals 1. What does all this refer to? Well, homozygous dominant is P times P, which is where your P squared comes from. This is represented right here. Homozygous recessive, remember we changed our little b's to q's. Q times Q, which is Q squared, that's represented by this individual. The heterozygous make it a little confusing. It's P times Q plus Q times P, which is simplified as 2PQ. And it's all set to equal 1 to represent 100% of the population. So let's look at this. Here's our equation here. Remember our common allele, the dominant B, is designated by the P here, which is going to be here, our homozygous dominant. Our less common allele, or recessive one, is designated as Q. And P plus Q must always equal 1 to represent 100%. A dominant allele B is for the orange color, and recessive allele will produce the white color. So individuals of homozygous for big B are represented here. These are our homozygous recessive for the allele. And this is our heterozygous, or one big B, one little b. So again, here's kind of relating that. Hopefully this will make it a little bit easier to understand, with our P representing our big B and our Q representing our little b. So we only have a choice between two, so that has to equal 100% of the population. And here we have our P squared representing our big B, homozygous dominant, Q squared equaling or representing our homozygous recessive, two little b's, and our heterozygous as big B, little b, and all equal 1. So a population of 100 tigers, 84 orange, 16 white, how many of each genotype exist is the question. So let's go through and let's calculate that in this box here. Well, remember our recessive, the 16 white, we know is going to be 16%, and we know what that genotype is. We can't assume we have a consistent amount for the dominant, because remember, it could be homozygous dominant or heterozygous. We want to start with the homozygous recessive. So 16% represented by 0.16, that's BB or Q squared. To get to Q, or just B, we have to take the 16 and square root it to get 0.4. Now we know that 0.4 plus x must equal 1, therefore 1 minus 0.4 would be 0.6. The dominant allele, or the big B, represented by P here, would equal 0.6. So what are the genotype frequencies? Well, we have to remember that we must assume this population is in Hardy-Weinberg equilibrium. So we just calculated with this information here the frequency of Q and the frequency of P, starting with the recessive. So to get to big E, which is P squared, we take our 0.6 and we square it to get 0.36. For our heterozygous, 2PQ. So here's our 2PQ. So 2 times 0.4 times 0.6 will give us the 0.48. And the last one here, our Q squared. We take our Q in this case, which is our 0.4, and we square it to get 0.16. All these added together should equal 1. So this gives you a little bit more of the background of the math here. Remember, Q is the 0.16, square rooted gives you the 0.4. P plus Q equals 1, therefore P equals 1 minus Q, which equals 1 minus 0.4, which equals 0.6. Again, might get a little confusing here. You're welcome to pause this at any time and look back. P squared is our 0.6 squared to give us 0.36, which means 36 out of 100 tigers will be homozygous dominant. We have the heterozygous here, which is the 0.6 times the 0.4 times 2 to give us the 0.48, which means 48 out of the 100 tigers will be orange and heterozygous. So again, take your time, go back, flip through this, and take a look at it. Assuming Hardy-Weinberg equilibrium, here's what we just calculated. This would be considered the null hypothesis. But now we take a look and we take some sample data. We take some genetic screens of our tigers. And we come up with these numbers. So how do we explain this data? Well, clearly the recessive ones match, but there is a strong favoritism, for whatever reason, for the homozygous dominant genes. So this may give these individuals a particular advantage versus being heterozygous and still orange. If we take another population, take a sample, we see here how to explain this data. Well, clearly the heterozygotes now have a clear advantage because they're being selected for. Because there's no phenotype difference, but this could be an advantage genotypically. Probably list a little bit more data. Again, always welcome to pause the video and take a look at it. This takes a look at allele frequencies. I'm looking at a number of individuals at 200 instead of just 100 here. Again, starting with the homozygous recessive because we know what that is, and you can go through and calculate that. In this case, the alleles contribute to a gene pool of 200. Remember, our genes are 200, but our total individuals are 100. Genes, because they're diploids, are multiplied by 2 in this case. And the possible gametes, you could see, again, follow this through. If you go back and watch the previous steps that I went through, this follows the same exact thing. And it's important to follow the math. And I'll also have another video going through another example to help provide you with some experience. I know initially this is a little confusing. Watching this again, doing some examples, as we will do in class, can definitely get you by that initial hurdle. And I think once you understand it, it'll come easier to you. Again, this shows the same example, a little graphical representation, adding in the Punnett square. So you can kind of see where the p squared comes from with the homozygous dominant, the q squared, which is the homozygous recessive, and then the heterozygotes here, and how these alleles break apart, and how they come up with the numbers that are calculated here. Last slide, just to go over in this presentation, we're looking at another example here. Again, tying in the Punnett squares, following the same equation of p squared plus 2pq plus q squared equal 1, and using now a different number of individuals, so you can see how we calculate the totals here. Individuals here is 500. We need to convert our number of individuals versus our total based on the 500 to get to our p's and our q's. And remember, p plus q equals 1. So by using genotype frequencies along with the known number of individuals of allele frequencies can be calculated, and using the allele frequencies here, predicted frequencies of homozygous, dominant, and recessive, and heterozygotes can be calculated. Remember, we want to start with the recessive because we know what those are. If we start with the phenotype here, we don't know if we're dealing with the homozygous dominant or the heterozygotes, but once we have the recessive, we know p plus q equals 1, and from there we can start to solve our equation and calculate the dominant phenotypes, what the exact genotypes are for those. So again, you can watch this video again. If not, I have another example video you're welcome to check out to hopefully get you through and understanding the Hardy-Weinberg equation.